Prostate cancer is a malignant enlargement of the prostate gland that occurs when some of the cells of the prostate grow abnormally. It is the third most common cause of cancer deaths. In Australia, more than 3,300 men die every year. Risk factors include age, particularly over the age of 50, genetic susceptibility, diet, higher intake of fat and meat. Current treatments are surgery, radiotherapy, and androgen deprivation therapy. Genetic damage results in some hallmarks that promote the progression of prostate cancer. Our treatment targets two hallmarks involved with the antioxidant system of glutathione, GSH. The first hallmark, resistant cell death, is due to DNA damage that causes increased reactive oxygen species, ROS, in cancer cells. In order for these cells to survive, they adapt by upregulating the production of GSH to regulate the redox balance in the cells. The second hallmark, sustaining proliferative signals, relate to the role of GSH in DNA and protein synthesis to promote cancer cell proliferation. Using the Worldwide Protein Data Bank, we will engineer a human cystinase enzyme that, upon administration, will bind to and degrade extracellular cystine of prostate patients to reduce GSH level. The synthesis of GSH involves cysteine, glutamate acid, and glycine with the assistance of ATP and catalyzing enzymes. The exact role of GSH in cancer is not currently known. What is known is that cancer cells experience higher oxidative stress from ROS than non-cancerous cells because of genetic alteration. Therefore, the maintenance of the antioxidant GSH is essential for their survival and proliferation. Under conditions of elevated ROS, endogenous cysteine production is insufficient for GSH synthesis. This stimulates the cysteine glutamate antibody to increase glutamate and cysteine uptake, which in turn upregulates the transsulfuration pathway. This pathway allows for increased production of GSH to a level that is able to combat the oxidative stress produced from the ROS and promote self-survival. Mitochondrial stores of glutathione disulfide, GSSG, are also able to contribute to increased GSH level.
After administration of cystinase, the transsulfuration pathway will be disrupted due to a depletion of cysteine. This will have a domino effect on intracellular GSH, resulting in a reduction of GSH and thus elevated ROS. The cancer phenotype will be affected as this enzyme causes immediate cell cycle arrest and death, and suppression of prostate carcinoma and tumour growth. Cysteine depletion also indirectly induces autophagy by the suppression of protein synthesis and upregulation of P-AMPK. The cystinase will be loaded using hydrophobic techniques into a biodegradable liposome, a micro-sized vesicular delivery system that allows for sustained drug release. This liposome will be composed of lipids that are covalently attached to polyethylene glycol, PEG, to increase its circulation time. It is known that the vasculature of tumours are leaky, allowing for the liposomes to infiltrate the cancerous tissue. But how will they only target cancerous cells? Upregulation of phospholipase A2 is characteristic of cancer cells in the prostate. Therefore, we have created a drug carrier liposome made up of phospholipid substrates known as pro-AELs. The phospholipase A2 that is secreted from the tumour tissue will hydrolyze the phospholipids, causing the breakdown of the liposome membrane, thereby releasing cystinase. The hydrolyzed AELs are cytotoxic to cancer cells and also help sensitize cancer cells for drug uptake. This can reduce the amount of cystinase that will be given by intravenous injection. Possible limitations that may arise from this treatment include cytotoxicity of activated AELs in the liver and kidneys. Therefore, we will need to monitor liver conjugation and renal clearance. Also, the invasiveness of intravenous drug delivery could be resolved by future study into an oral form. Thank you, and please fund our preclinical trials.